So our next speaker will be Dr. Deng.
If I talk 30 minutes, then you don't have time.
Okay, so I don't have time to introduce him then. And Dr. Deng is also a faculty member at the Les Turner Center, and we are very happy to have him with us.
Okay. I'm going to talk about something, our recent work, which has not been published, to show the efficacy and the long-term safety of the CRISPR Cas9-mediated SOD editing in ALS. So 10% about 10% of ALS are inherited or has a known genetic mutation underlying the disease. And among this, about 30 some genes that has a strong linkage to causing ALS. For about 60% of them, 60% of the familial ALS can find a genetic mutation in European or we say Caucasian ALS patients. Because this one, C9 is the most common, and so in the worldwide, the SOD1 probably is the most prominent, and it's far more common than the other genes linked. So in the about 10% of the familial cases may have genetic mutation, like in SOD, C9, and TDP43, and the fuse. These four are the most common. So in 1993, we found the genetic mutation in SOD1. And in 1994, we made the mouse model. And since then, we try to understand how the mutation in SOD1 causes ALS and try to develop therapeutic treatment. And we thought, because SOD1 is a free radical scavenger, so we thought this mutation may increase the free radicals instead of remove the free radicals. So we thought if we increase the SOD expression, that will increase the removal of free radicals. That should have therapeutic effect. So the hypothesis looks very reasonable. So we made the mouse model and overexpressed wild-type SOD1 and the ALS mouse model. And what we found, actually, the result is opposite. Overexpression of wild-type SOD1 not slow down the disease. Conversely, it exacerbates the disease. For example, normally the mouse will develop disease around 100 days. And if you put wild-type SOD1 there, they will develop disease on 80 days. And the mouse die much earlier. And the most interestingly, the AFV mutation never develop disease, the mouse model, because of low expression. But if you put wild-type SOD1 there, the mouse will develop disease and die earlier. And the other one is the same. Normally, we have this mouse model, and they will develop this around 300 days. And if you put wild-type SOD there, the disease is about 180 days. So that raises the question, why the mutation of SOD causes disease, and how the wild-type SOD1 exacerbates the disease. And eventually we found the wild-type SOD1 was recruited by the mutant SOD1 into the protein aggregates. And these aggregates are intermolecularly cross-linked through disulfide bond. That means if the wild-type SOD1 is there, then the mutant SOD1 can recruit the wild-type SOD1 into the aggregates, and these aggregates are toxic. So based on this result, and so we know the wild-type SOD1, even though it's a function to remove free radicals, but it's a substrate. So the substrate, if the substrate is recruited into the SOD aggregates, it's toxic. So you cannot use SOD1. But any other antioxidants may be useful. Notably, there's a drug called Davee, and that has been tested in this mouse model and showed some mild effect in 2008, and this drug has been proved in last year. But notably, the therapeutic effect is very mild, and that suggests only antioxidant alone in the patients may not have a dramatic effect, therapeutic effect. Most likely, it's the intracellular free radical, or we say redox homeostasis, is so difficult to manipulate. So currently, there are many. So that work means if we either reduce wild-type SOD1 or mutated SOD1 or both, we have a therapeutic effect in the patient, the SOD-linked ALS. And currently, there are many types of drugs or compounds have been tested in the animal model and showing a variety of effect. And some of them are in clinical trials. And the small molecules, anti-oligos, and the HSP inducer has been almost completed in the trials. But so far, there's no therapeutic benefit has been shown in these trials. Of course, the dose may be optimized, and the way of administration may be optimized, and also the sample size will be increased. But that also suggests, and these therapeutic strategies may not be that effective, meaning alternative approaches we may need to explore or test it. And obviously, the CRISPR-Cas9 is one of the alternatives. And in the mouse models, for example, SOD1 mouse model, it's a very robust pathology and phenotype. For example, they develop disease around 100 days, and they will die like 130 days. So if some therapeutic approach is really effective, it will be shown in this mouse model. So we tried this one. And CRISPR-Cas9, everybody knows it's effective, but why the clinical trials are very hasty? Because there's a major safety concern. And once off target, that means you're not only targeting what you want to target, they also target some other places. So that may raise the safety concern. The other one is large deletions. They may, at the targeting site, they may lead to the very big DNA fragment deleted. And we don't know what is the mechanism, and whether these are controllable or not. And if we understand the mechanism, we may use this mechanism to avoid or minimize this effect. The third one is non-term safety. And most of them, when you treat the mouse model, they observe the phenotype for a few weeks or a few months. And the problem is also, targeting efficiency is very low. If targeting efficiency is low, then the observation is short. So there's no convincing data to show it's really, really safe. For example, obviously there's a cancer risk. And for example, in the P53 knockout mice, it takes like half a year to develop lymphoma or sarcoma. And also for liver cancer, it takes maybe 1.5 years. So to address this concern, the safety concern, we designed an experiment. We completely target the gene we want. And then we observed the phenotype and the pathology for lifespan of the mice. That means over 2 years. So this data has not been published, but anyway, I would like to show this information. So we designed a target and target X2 and made the transgene and put in the vector and cut this one to make the mouse model. That means this mouse model will express the Cas9 specific sequence, target the human SOD, and the Cas9 enzyme that will edit the targets we want. And when we design this strategy, we try to optimize the sequence, and so there's no highly homologous fragment or DNA sequences in the mice, or in the human genome. And that only 3 sites with 2 mismatch, and the humans at least 3 mismatch. So theoretically, it should be safe. We made the mouse model, and then we crossed with our GYH mice. Normally, the GYH mice, we can see the control is 100 some days. They will develop disease. They will die around before 150 days. But in the in the mice we treated with CRISPR-Cas9, they never developed disease. We have 50 mice, and the 50 mice over 150 days, none of them developed disease. And two of the mice is now 800 some days, so nearly 2 years. So we feel this one looks like it's very efficient, at least prevent the development of the disease in the mice. So we analyze the pathology. We can see this is the untreated mice, and this is the treated. We can see the protein aggregates with SOD and the ubiquitin are gone. It's not present at all. And also the GFAP and astrocytes and the microglia markers are as normal. And also the neurons in this one is normal compared to with this sick mice. And the nerve root is normal in compared to the shrinked one. And the muscle is normal. So the Cas9 in this system, even though you expressed from the very early stage, in the lifetime, actually we didn't find anything like a tumor or inflammatory disease. And these mice really do not develop disease anymore. Yes, I want to show this video. How can I show this video? Oh, so you can easily tell which one is treated, which one is not treated. You can see this one is treated, and this one is not treated. It's you can see the leg. It actually is start to paralyze. And then this mouse died 2 weeks later. So that means the phenotype and the pathology-wise is really completely prevented by CRISPR-Cas9 treatment. And then we analyzed whether the SOD1 is really, the transgene has been really targeted. And then we sequenced this many, we found 19 types of ADs in the SOD gene. And this is the summary. You can see they have a different variety of deletions or insertion. And then we can see SOD, this is the not treated. It's normally over 10 times of the endogenous. When you treat it with Cas9, it's completely removed. Only this one left is the mouse endogenous SOD. And the GFAP becomes normal, and the microglia marker is normal. And so it's really, the targeting efficiency is really high. And this is another one, the mouse model we analyzed. These how many, more than 100 clones. There are different targets with this copy. So what is the off target? Yes, because when we designed the experiment, we try to avoid the off targets. But anyway, you still have something, there's a risk there. For example, this is the 10 top candidate of the off target. And for example, and this is a score. Even though people that try, this part is most important. The C-terminal part, the 3'N part is most important. And the people in the cell culture system have been saying the 12 base pair is most important. If you have some mismatch, and the 3'N 12 base pair close to the pair should be okay. But if you don't have a mismatch, that is risk. So we analyze all of them. So we sequence clone DNA fragment and into bacteria and analyze individually. We analyze more than 100 clones. So that means if I have a target, about 100%, we should be able to detect. Eventually, we found only one. Potential of target number 2. We have found 3 clones. And interestingly, see this one, that's actually 14 base pair, 14 nucleotide, no mismatch. But number 7, we have 15, but you still have a mismatch, but you don't have any mutations there. So suggesting,
Here, we get tweets from patients, and they cannot hear you. Is it possible for you to speak to the microphone directly?
Okay.
And it says the voice is coming and going.
Okay.
Can you speak to the microphone?
Yeah, this one?
Yes.
So we can see the only difference, one is AGG, one is GGG, suggesting GGG probably has higher targeting efficiency. So when we design the experiment, we try to, if it's off target, we try to avoid GGG. And this is the individual variations we identified. So meaning the SOD is 100% targeted, but off target is really rare. And if we take this one, it's only 3 here, 3 in the second mouse. So less than 1%. Okay, the second one is large deletions. And yes, we found deletions, large deletions, depends on the size. And almost 50% of the targeting events are large deletions. And this is unexpected. And it has not been reported like this often. So we try to understand what is the problem. So we cloned the 6 kb fragment and try to analyze what are these deletions. And eventually we found two deletions. And for example, the DNA fragment, there's a crossover here. And maybe short DNA fragment, about 23 some base pair, and 30 some base pair deletions. See, because this one is identical. So we put a name there called, there's no description for these events. So we put a name, proximate, and it means very close, 2 identical sequence, very close. So we call it proximate identical sequence, mediate large deletions, not large deletions. And because they're identical sequences, so we searched all the SOD DNA gene there, and we try to predict whether these deletions are predictable if we use proximate identical sequence. So we identified 15 types of deletions. Then we sequence. And yes, we identified this many, 8 types of deletions that are all caused by the proximate identical sequence. See, these are sequences identified. And because there's some other deletions we cannot detect, so we suspect this is intra-arm deletion. That means on one side of the arm. And we predicted 14 types, and eventually, yes, we identified 2 of them. 1, 2, 3, 4 of them. So all the 12 so-called large deletions are all predicted by our, based on our knowledge. So that means these deletions, or so-called large deletions, people previously are so concerned about the consequence of these deletions. Actually, they're all predictable. So we can use this knowledge to design, to optimize our therapeutic development. See, based on this one, we can see all deletions. How many? 50. More than 70%, more than 70 types, more than 70 deletions. 1, 2, 3, 4, several types. And we can say, so then we summarize this data. What we found is the events occur that is correlated with the size of the proximate sequence. If you have small sequence, if you have a big sequence, then you will have a higher frequency. The second one is the distance between these 2 sequences. If far away, then the frequency is very low. If very close, it's very high. So this one we can use this knowledge to design our experiment, how to target, how to efficiently target, and how to design a safe experiment for treatment. So overall, I summarize this one. So our data suggests the CRISPR-Cas9-mediated genome editing is a very effective approach to target the disease gene. If you have disease gene, for example, SOD1 or something else, if you know it is a gain-of-function mutation, and you can use this one to target. And this one provides preclinical evidence for therapeutic development. The second one is, in our sense, we test this model, the SOD1 in our mice over 2 years. And actually, the longest lived mice is 34 months, almost 3 years. And we did not see any cancer or inflammatory disease. Suggesting it's reasonably safe. Remember, the SOD1 FOV mutation, which accounts for half of the ALS cases in North America, the survival is only 1 year after the onset of the symptoms. And now we observed at least for 2 years. Anyway, we have not seen anything in these mice. The third is the off-target deletions and large deletions are largely predictable. Anyway, 12 types of mutations are all predicted through our system. And so when you design the experiment, you know what will happen. So you can use this knowledge to predict and optimize your study. The third one is, so when you design the study, if it is possible, then to avoid the identical sequence, which has 15 nucleotides, then you will diminish the possibility of the deletions. If it's not possible, if not avoidable, then remember, the rate of the events is positively related with the size of identical sequence, but negatively related with the distance between them. Then choose a small fragment, choose the small identical sequences, and with non-distance. That way, then you will be reasonably safe. I think I just stop here and take questions because I have leave 5 minutes for questions.
So if you want, you can actually have...
I think it's okay, you know. I finished. Yeah.
Hey, Deng, I think your data is really promising. But essentially, you took a mouse model which has between 25 and 35 transgenes of SOD1, and you used CRISPR-Cas9 at the embryonic stage to delete those transgenes. So how do you think this will apply in a situation where you have to target the endogenous single copy of SOD1? The reason why I bring that up is because there's plenty of evidence showing that the efficiency of CRISPR editing has a lot to do with DNA context, how available the gene is, how highly it's expressed, and things like that. So how do you think this applies, what you've learned applies to targeting the endogenous gene?
So this one, the design of this experiment the purpose of this study is to address when the gene is effectively targeted, what is the side, let's say the side effect, whether it is safe. So what we want to say is, even though we have 20-some copies, actually we determined there's 30 copies of the transgene, they are all targeted. We observed for 2 years. Then actually we did not see the major side effect in the mouse system. Of course, in the human system, you have only a single copy. So theoretically, it should be easier. It's much easier than the 30 copies, because the 30 copies are all targeted. So in the humans, when you're in the clinical trials, you may use the AAV system, and this one AAV system has been proved for the Cas9 AAV system has been proved for clinical trials in retina with the so-called Leber's congenital the pigmentosa. So, of course, that one is only locally rejected. But if that one is successful, then we can also apply maybe eventually to the central nervous system. I think our data is provided the preclinical evidence to show the safety after in the condition when all the genes are targeted, whether it is still safe.
So can you update us on where we stand with Cas9 or variants of Cas9 to be packaged into viruses? I mean, I think the problem is the size. Maybe you could tell us where the field is.
Yeah, because in the clinical application, so the Cas9 there are several systems, and what we use is the SP system. The SP system is the protein. The Cas9 is 1,300 some amino acids. So the coding cDNA is more than 4 kb. So we know the AAV system only accommodates 4.7 kb, the maximum. So that means one single AAV can only accommodate a single SP Cas9. So then you have another one. You have a guide RNA. So you have to pack it in two AAV vector. There's another one. It's called SA system. SA system is about 1,000 kb. So it's about some kb the cDNA, and then you include the guide RNA and this one. So that one can be a whole thing can be accommodated into the same virus. We also use that one. We try to use the small vector, but the promoter has some problem. The efficiency is very low. We're still optimizing that system. So, but anyway, if you want to target a single guide, a single site, I think the SA Cas9 should work. But if you want to target the multiple site, and like currently the clinical trials, the retinitis pigmentosa, they want to target both sides, that cut the fragment out. So that one, only SP probably, only the two system, we have to use two systems, two vectors. A single one won't work.